The first type of anchoring junction we are going to talk about are adherent junctions. So let's start with that. Adherent junctions, they can have different forms. In non-epithelial cells, adherent junctions are in form of small punctate or streak-like attachments that indirectly connect the cortical actin filaments of two interacting cells or two adjacent cells. In epithelial tissue, continuous adhesion belt, which is also referred to as zonular adherens, just below the tight junctions forms encircling each of the interacting cells in the sheet. So epithelial cells, they form a sheet. The adjacent cells, they have this network of actin, which basically is just below the tight junctions, and it is basically encircling both the interacting cells individually. Adherent junctions, the adhesion belts, are directly opposed in the adjacent cells in which the interacting plasma membranes are held together by cadherin molecules. Cadherin molecules are a type of a sticky molecule that are responsible for forming cell-to-cell junctions. We'll talk about cadherin molecules in the later module. But here I just want to mention that these cadherin molecules, they serve as the transmembrane adhesion proteins, which are basically different domains. They have extracellular and also intracellular domain, and the extracellular domains of two adjacent cells, they glue together with each other, forming this type of junction. Adherent junctions in a form of adhesion belt, you can see them in this photograph here, for example. These are the belt-like junctions. Basically, if you have to think about these cells in 3 dimension, that these cells have 3-dimensionality. So individual actin filaments are basically encircling the top part of the cell. So it is basically a ring. And these rings of adjacent cells are connected to each other by cadherin molecules. So here is, for example, lumen. We are looking at small intestine, epithelia of small intestine. So here's the lumen. This is the lumen. So here you can see that some of the actin filaments also move inside the microvilli. We talked about that briefly in the previous lecture. So additionally, I would also like to mention that you can see tight junctions here. Formation of tight junction requires that first adherent junctions form. So if there are no adherent junctions, tight junctions cannot form. So basically, this is the structure of cells linked together by adherent junctions. Now we are going to magnify that region we saw in the previous slide where there was a blue box, and look at these cadherin molecules that are holding these two cells together. So cadherin molecules, they have these domains which stick outside the cell, and part of their domain is embedded in the plasma membrane, and the other domain is actually basically inside the cytoplasm, where there are a set of proteins. These proteins are called the anchor proteins. Basically, they're anchoring the cadherin molecules to the extracellular matrix, to the cytoskeletal elements. In this case, actin filaments are shown, which are responsible for adhering, which are responsible for their interaction. In case of adherent junctions, actin filaments interact with the cadherin molecules. We will see in a different type of junction. It is intermediate filament, for example, in desmosome, which interacts with the cytoplasmic domain of cadherin molecules. So here's cell number 1, here's cell number 2, here's the plasma membrane. So as you saw earlier, that these actin filaments are forming sort of a circular belt at the apex of the cell, towards the apex of the cell. And so the adjacent cells are basically linked together. Their actin filaments are linked together. This is a transcellular linkage of actin filaments with the help of cadherin molecules. Cadherin molecules, these are the sticky molecules, as I mentioned. They form homodimers, meaning two similar cadherin molecules can stick with each other in the plasma membrane of each interacting cell. So here, for example, is a cadherin molecule in the form of a dimer. These two cadherin molecules belong to this cell. They are sticking with the cadherin molecule dimer of the adjacent cell, thereby linking these two cells. So this is the basically interaction of the cadherin molecules that is linking or hooking up these two cells. The intracellular domains I mentioned already, they anchor proteins, cadherin proteins to anchor proteins, and these anchor proteins in turn tie them to actin filaments. Actin filaments are joined, basically if you think about it, the actin filaments, this transcellular network of actin filaments is joined from one cell to the other cell to the next cell into all the cells present in that particular tissue. Here, another thing I would like to mention, an important role of these type of junctions, adherent junctions, is during development. So we know that during development, different tubes have to be formed. For example, neuronal tube has to be formed from where our nervous system of the vertebrates basically forms from this neuronal tube. So how do you form a tube from a sheet of cells? Is basically here you have epithelial cells linked together. Here you can see the red is basically that transcellular network of actin filaments that I talked about. So if these actin filaments we know can slide against the myosin filaments, that happens in muscle contraction. So if there are myosin filaments also present, which there are, these actin filaments, they slide against, when they slide against the myosin filaments, they basically pinch, or this pinching force basically results in invagination of some of these cells. So there's a contractile force in this direction, there's a contractile force in this direction. The end result will be formation of this loop or this invagination you can see over here. Ultimately, through other changes, these cells will basically disengage from epithelia, and they will form a different type of group of cells, which ultimately will give rise to, for example, neuronal tube. So there's a lot more happening here. There are other, of course, sticky molecules that dictate how these cells can move out of this particular sheet of cells. Let me demonstrate this phenomena with the help of a little cord. So here we have a cord. If we apply force, if there's a contractile force which is applied here, meaning that inward force, it will basically result in formation of this loop. Ultimately, what happens is this loop will come off, it will detach from this sheet, and it will become an independent entity. This is how basically the neuronal tube forms during the development. We will continue our discussion of anchoring junctions in the following module.